Welcome back. Today we will take a look at gel permeation chromatography, also known as size exclusion chromatography, which is a technique used to separate and analyze polymer samples based on their molecular size or hydrodynamic volume of the analyte. GPC is extensively used to characterize micromolecules such as natural polymers, synthetic polymers, or proteins. The measurement process involves passing the mixture dissolved in the mobile phase through the stationary phase. Separation occurs via the use of porous beads. Stationary phase is depicted here in gray. Here we can see the molecules of three different sizes moving through the column. The rate at which the sample components travel through the stationary phase depends on the difference in hydrodynamic volumes between the individual components. The smaller analytes, shown in red, can enter a higher fraction of the pores and therefore spend more time going through the column, increasing their retention time. Conversely, larger analytes can enter a smaller fraction of the pores and are eluded quickly, meaning that they will exit the stationary phase at different times. Now we can take a look and see what happens inside the column. The green band containing the larger molecules moves faster than the red band, which contains the smallest molecules. Thus, the molecules are separated based on the hydrodynamic volume. Let's now take a look at what the detector sees. Let's plot intensity of the signal against time. When a band passes through the detector, it registers an input. Of course, as there are a lot of molecules, they can act slightly differently, leading to some peak broadening. Again, we can see the order of the molecules based on the size. The retention time is the time from sample injection to the peak maximum of a component. To account for system delays, an adjusted retention time is often used by subtracting T0, the time it takes for the mobile phase to pass through the column. This allows calculation for the retention factor, which describes how long a compound is retained relative to the mobile phase. Molecules that are larger than a particular size, which is known as the exclusion limit, are not able to enter any of the pores of the column and will elute together with the solvent. The elution volume of these analytes will be the same as the void volume of the column. You can see the exclusion limits of different columns on the screen now. The unit here is Daltons or grams per mole. The permeation limit is defined as the maximum size of a molecule that can enter all the pores of the stationary phase. If a molecule is smaller than this limit, it will penetrate all available pores and elute together with other small molecules at the same time, regardless of molecular weight. The GPCs at the department are equipped with two types of sensors. The refractive index detector uses a light beam passing through a dual chamber flow cell, one side with pure solvent, the other with solvent and sample. Differences in refractive index between the two sides bend the light, shifting it towards photodiodes. The signal's direction, either positive or negative, depends on whether the sample has a higher or lower refractive index than the solvent, and its amplitude reflects both the sample concentration and refractive index increment. The UV-Vis detector measures light absorption by the sample. To generate a signal, the compound must contain a chromophore, a part of the molecule that absorbs UV or visible light. Detection follows Beer's law, meaning the signal is proportional to concentration. In this system, a photodiode array captures the full absorbance spectrum, allowing detection across multiple wavelengths at once. Let's now move on to sample preparation and measuring. Once your sample is ready, weigh the appropriate amount on a scale. The concentration ranges for each molecular weight are shown on screen. Additionally, the shake times for each molecular weight are shown on screen. Then we want to dissolve it in the appropriate solvent. Keep in mind the solvent used will be written on the machines themselves. We now place our sample on a shaker, or in our case, a magnetic stirrer. Now we use a syringe to gather the solution, and now we filter it, because we want to remove any particles present. Even the smallest particles can cause blockages in the GPC tube, so it's important to avoid this. We filter our solution into a GPC vial, which is shown on the screen. These are small vials and their lid has a more flexible silicone region, where the needle of the GPC can extract the solution. We also want to create a blank solution, as this allows us to test the GPC performance and see if any irregularities are present. Make sure to also label your samples, as otherwise they might get mixed up. We then place our GPC vials into the trays of the GPC. Remember the number of the tray and the position of your vials, as they will be used later on in the software. On the computer, go to the Real-Time Analysis window. On the left from the main tab, open Real-Time Batch. From this icon, open the project folder you want to use. Now we want to set the appropriate vial number together with the tray number. You can find these on the tray itself. Also, label the sample name appropriately. Choose the appropriate method for your measurement. Select the data file where you want your measurement to be stored. Now we go back to the Data Acquisition tab and check if the settings are correct. If you want to use different settings, you need to create a new method, but usually we just want to check if the flow is 1 milliliter per minute. Then we start the measurement by clicking Start Real-Time Branch. The GPC will now take a measurement from the vial using a syringe. If we open a panel, we can see that there is a pre-column to protect the actual GPC column. Then the liquid goes through the main column. After the measurement is finished, we want to go to post-run in Lab Solutions and then GPC post-run. We open the location of our measurements and then we can see the results. We also need to load the appropriate method parameters for the calibration. Choose the most previous one. We now want to manually delete the peaks which are caused by the solvent. By clicking Data Report, we can generate a PDF of the results. We have to select a report format file, which makes the report in a nicer format. We then select File, PDF output and save it as a PDF. We can also save it as a text file by going to File, Export Data. And there we can select the wanted parameters. Click Output File and set the correct name and location. This data can be then plotted using software such as Origin or Excel. For accurate interpretation, it's helpful to know the molecular weights of all components in your sample. In our case, we only had one substance, so this was not an issue. It's also important to note that the refractive index detector is positioned after a UV-Vis detector, which may cause a slight delay in elution time for the refractive index signal. Let's take a look at a more complex sample. Here, oligomer 3 is shown in blue, plotted from the raw data exported from the instrument. We can fit individual peaks to match the overall shape of the oligomer distribution in order to determine the relative fractions of each component. For accurate results, it's essential to have prior knowledge of what your sample contains, as this guides peak assignment and interpretation. Thank you for watching the ChemTube video on GPC.